The overall goal of this method is to amplify 16S rRNA from human milk and other low biomass samples using a semi-automated protocol.
This method can help answer key questions in the development and perturbations of the human microbiome as it relates both to health and disease.
The main advantage of this technique is that it's semi-automated, relatively easy to perform, and adaptable to multiple sample types, including secretions, swabs, and stool.
After thawing and aliquoting whole milk according to the text protocol, spin the samples at 5,000 times G and 4 degrees Celsius for 10 minutes to pellet the cells. Use a plastic spatula or large bore pipette tip to remove the top fat layer. Then, without disturbing the pellet, remove all the supernatant except for 100 microliters. With 1 milliliter of PBS, resuspend and wash the pellet. Then prepare a negative control by adding 1 milliliter of sterile PBS to a 5 milliliter tube. Transfer the suspension to a clean 1.5 milliliter centrifuge tube and spin it at 5,000 times G at room temperature for 1 minute. Then use a 1,000 microliter sterile filtered pipette tip to discard the entire supernatant fatty layer. To disrupt and homogenize the sample, add 600 microliters of lysis buffer containing beta-mercaptoethanol to the pellet and transfer the suspension to a bead tube. Prepare one positive control with lysis.